In our project, we worked on the stem cell differentiation protocol database. We worked on a prototype of a website that was provided by our client. The goal of this website is to collect all of the different stem cell differentiation protocols. Currently, these protocols are scattered all throughout different scientific articles, and there's no consistent way of noting them. Therefore, the goal of this project was to create a database that would collect them all in one place and show them in a similar manner. On the main page, we implemented the counter of the participants, papers archived, and the total number of protocol submissions. The protocol upload page was already created by our client. A user can upload an article. A feature that we implemented is this visualization tool on the side of it. These will automatically show up. This can be submitted. However, to submit a protocol, a user has to be logged in. The visualization can also be exported and downloaded as an SVG file or a JSON file. A user can sign up with their first name, last name, affiliation, email address, and motivation. The admin of the website, which is our client, receives an email and they can approve a user. Here, a user can see all the different protocols that they have submitted. They can either edit them, delete them, or make them private or public. Next, we have the query tool. Here, the user can search different cell types. The query tool shows all the entries in the database, including their article number, their PMID, which upon clicking redirects to the protocol page. They can also see the specific protocol that has been submitted. Within the visualization tool, users can see the different growth factors that have been used across time. So time is on the horizontal axis, and here we can view different growth factors that have been used. All the entries can be filtered by publication year, number of steps, number of days in the protocol, starting cell type, cell line, sequencing data. There's an additional tool that allows users to compare different protocols. We can also click on different growth factors and see where they are. We can once again either see the article or the protocol, and we can also export like so. The next feature is the lineage tree. It visualizes the different differentiation pathways. It starts from undifferentiated pluripotent stem cells, and then it diverges into different cell type. A user can search up a specific cell type, like so. Upon clicking on a node, a user can see all the matching protocols, the growth factors that have been used, as well as the readout, which are the gene markers that have been used. A user can also click on a particular node. Here, we clicked on a node that connects pancreatic progenitors with pancreatic endocrine progenitors. Here we can see all of the matching protocols. The blue branches correspond to matches. These are the branches in the tree that have matching protocols in the database. The same goes for nodes. The green nodes have matching protocols, while the white ones do not.